Okay, thank you everyone for joining us today. We have another Some Talk next week as well, so please return. And actually, the name just left my head, so I'll check it for after. But today, we're very pleased to have Will Hendricks visit us, and he is an adjunct professor at TGen, also with leading a new startup, Vidium. Did I say that right?
Vidium, yeah. Vidium was the other startup. There's a new one I'm working on now.
Oh, there's a new one. We're going to get the skinny on it all, which I think is really exciting to hear about, kind of a little bit outside the academic world and how that works. But so without further ado, actually, I'm just going to let him begin, and I will run around the room at the end to help with questions.
Awesome. Sounds good. Thanks, Ann. All right, folks. Well, thanks for having me. It's exciting to be here and looking forward to hopefully some interactive discussion with you folks. I think as you can see from all the titles that I'm collecting over here, I'm actually a bit of a mercenary these days. I've spent most of my life in my professional life in the academic translational research world, but over the past 7 years or so, I've moved from doing discovery translational science into actually the startup side of life. I had a lab at TGen for about 10 years that was focused on comparative oncology. We then built a startup inside of TGen that Ann was referring to called Vidium that was doing cancer diagnostics for pet dogs. We sold that company about a year ago, and since then I've been floating around doing lots of different things, spanning venture capital, fundraising, to consulting for other startups, to actually dreaming about building the next startup that I actually want to talk to you guys a little bit about at the end and get some ideas, because it's very early days in that next startup journey. So maybe a different kind of talk than what you normally have, given that spectrum of interest and that lens that I have on the scientific process. I think at heart, I've learned over the years that what I'm really most interested in at heart is bringing products to market. So that translational science piece, which is trying to bring discoveries to the bedside. Once you actually take that out commercially, that's part of what I really get excited about as well. So sort of the end of the scientific discovery journey. So I'm going to be talking about the state of genomic medicine and veterinary oncology specifically. That's really my main focus and where my research and passion lies. We're going to have a nod to human oncology in there. And we're going to be talking about some of the big problems that exist in veterinary oncology today and how we're trying to solve some of those problems. And we're going to do this through the lens of cancer genomics, comparative genomics, and the evolutionary forces behind cancer. So let's start with just stage setting on what are some of the major issues in vet oncology today. How many folks have pets at home? Lots of folks have pets at home. Probably many of us, unfortunately, have experienced all sorts of disease in our pets and have experienced the difficulty of actually working through having disease diagnosed. Disease management can be a very challenging process to manage, especially cancer in its own right. We have a shortage of specialists in veterinary medicine in general, and that's certainly true in oncology. There are only about 420 veterinary oncologists in the US, which is a big discrepancy between the number of GPs and oncologists. So if your animal presents with some strange symptoms, maybe coughing, maybe lethargy, even worse if they are crashing out and you take your pet to the ER or to the GP, it can often be a very long process to even have cancer diagnosed in your pet in the first place. Once it is diagnosed, there are very long referral times to actually see an oncologist. There are a few in the Phoenix area. There's a group of them in Tucson, but you might have to wait 3 months between actually identifying a possible cancer case with a GP and actually getting in to see an oncologist. Once you do get in to see the oncologist, the treatment options for your pet may then be very limited. The standard of care for dog cancer, and even more so for feline cancer, is vastly underdeveloped relative to human. So we have a lot of gaps in the care continuum for pets with cancer today. We're definitely not finding cancer early on the screening side of the equation. There are new and emerging screening tools for dog cancer in particular, but it's still early days there. Diagnosis and treatment are difficult. We don't have a lot of great tools on either the detection or the drug side of the equation. And then once you do have an animal under treatment, disease monitoring is challenging as well. And yet there are 4 to 6 million cancer diagnoses in pet dogs in the US every year. And cancer is really the #1 killer of certainly older dogs, but probably much more broadly across the spectrum. Epidemiology in veterinary is also not fantastic, but cancer is a big problem in dogs, and we don't have a lot of solutions, in short. If we look instead at what's happened in human oncology, there is a stark difference between the amount of progress that we've made. Certainly, there is a lot that we still need to do in human oncology, but if we're looking over from the veterinary side of the equation, we look over with a lot of jealousy. We have reduced overall cancer death rates by more than 33% in the past 20 to 30 years. A lot of that is due to innovation in screening, but also in treatment. And a lot of that is on the foundation of our genomic understanding of cancer. If we look at specific examples, like the example here of melanoma, metastatic melanoma 3-year survival over time, and how that's changed from 2000 to 2018. If we look at this little curve right here for disease that is essentially metastatic, it's distant at diagnosis, that relative survival went from being 20% of patients at 3 years in 2000 to this bump around 2010, and we've now doubled that, and it's continued to go up over the past 5 or 6 years as well. It's a pretty remarkable difference. We don't have examples like this in veterinary oncology. And that inflection point, for those who are in the know, actually is related to the development and deployment of targeted therapies for metastatic melanoma patients. Now we have immunotherapies as well. Our understanding of the genetic underpinnings of melanoma was evolving around here. Our first targeted therapy for BRAF mutant melanoma was deployed right around this space, and that's when we started to see these significant improvements in survival. So much of this progress is due to the precision medicine revolution that's happened in human cancer, and that is just starting to make its way into the veterinary space as well. So cancer is a genetic disease. Let's get centered on cancer as a genetic disease. That's a lot of how I'm going to be talking and thinking about cancer. We know that cancer arises through the stepwise accumulation of mutations in genes that control cell life and cell death, shown very schematically here. As we accumulate those mutations that confer malignant phenotypes on a growing tumor population, eventually cancer will invade surrounding tissue, that will spread to distant sites, and it is that cancer metastasis, as the cancer spreads, that ultimately usually kills cancer patients. Those mutations all converge on these different phenotypic hallmarks of cancer, from sustaining cell signaling to evading growth suppression, avoiding immune destruction, et cetera, et cetera. So these mutations hyperactivate or cripple key pathways that regulate these phenotypes and eventually enable the systemic progression of disease. Where do mutations come from? This is important in how we think about how to manage cancer as well, whether dog or human. There are endogenous sources of mutation. There are inherited sources of mutation and exogenous. Endogenous sources of mutation are actually the most abundant, just by virtue of cells growing and dividing. Every time a cell divides, errors are made while DNA is being replicated. Sometimes those mutations can confer a fitness advantage on the cell. They occur in oncogenes or tumor suppressor genes. Of course, we're all familiar with a lot of the exogenous sources of mutation, UV, for example, in sun-exposed melanoma, cigarette smoke and lung cancer, even pathogens cause cancer. About 90% of cancer is actually sporadic and not inherited. About 10% of cancer is actually inherited. So most of the mutation burden that is driving cancer is actually happening in this bucket, and a lot of it actually is happening here and across these different domains. This is important, again, as we start to think about how the natural history of cancers in humans versus dogs, and we'll talk a little bit more about that. So why do we care about mutations in cancer? Well, as the foundation of how cancer develops, they also comprise the key biomarkers by which we can diagnose cancer, but also the key targets, or at least the directors of the pathways that are targets for the treatment approaches that we take to cancer. We've got a few examples here of how cancer mutations can be used in different diagnostic settings. The classic example of BRCA mutations in breast and ovarian cancer and the predisposition to those diseases, so they can support understanding the mutation status of BRCA can help us understand risk profiles for cancer patients and cancer in families. Mutations are also used diagnostically. One of the great examples here is the Philadelphia chromosome, the classic translocation of BCR and ABL that occurs in CML. This occurs in about 95% of CML patients and can be used in the differential diagnosis of CML amongst other disorders. That mutation actually also discovered in the 80s and 90s drove one of the first targeted drug development programs and resulted in the drug Gleevec that has transformed the care of CML and CML patients. That was an example of one of our first targeted therapies based on the discovery of a mutation, the development of a drug that could just hit that mutation in the cancer cells and not the normal cells. That has been a great tailwind going into the 2000s and the genomic era that actually has helped us realize the value of that approach. We're actually lacking a Gleevec moment in veterinary oncology, and we have some challenges because the drug development history in veterinary oncology has not had that sort of tailwind as well. So I'll talk a little bit more about that too. So a little bit of a tangent, but that's a particularly important mutation to reflect on, both for diagnosis and targeting. And then we have examples of mutations that can be used in prognostication. For example, p53 mutations in breast cancer can predict poorer survival in patients with a mutation in the red survival curve here versus the wild patients with wild type p53 in blue. p53 is not often used in prognostication because there are a lot of other markers in breast cancer that are a lot more powerful than p53, but just an example of how, again, mutations can be used to predict, to manage risk, diagnose, and prognose in cancer patients. Of course, as we just talked about with Gleevec and the Philadelphia chromosome, mutations also serve as therapeutic targets that selectively occur in cancer cells and not in the normal cells in the patient. So before the era of targeted therapy that, again, was sort of ushered in by Gleevec and the Philadelphia chromosome and then the genomic revolution, much of cancer chemotherapy was based on the use of cytotoxic drugs, which essentially are broadly toxic drugs with a very narrow therapeutic window that are really good at killing rapidly dividing cells. So these broad toxins that kill rapidly dividing cells like cancer, but also cells in your gut, lots of other normal cells in the body as well, thus the toxicity of chemotherapy. But that was the main set of tools we had in terms of chemo until we had targeted therapies that have a much broader therapeutic window because they are selective for mutations in cancer cells. Another great example of one of these mutations is, I showed it earlier on, it's in melanoma, in metastatic melanoma. So we discovered probably around, we the community, this isn't me, the royal we, I guess, discovered around 2005, 2006 that about half of metastatic melanoma cases are characterized by mutations in an oncogene called BRAF. They occur in a single site in BRAF. They drive activation of the pro-growth MAPK pathway. They help melanoma cells grow out of control. Melanocytes turn into melanoma cells and grow out of control. This is an example of the MAPK pathway here. That discovery, it was actually before we were doing broader genomic profiling of cancer. There was still targeted studies by which we found BRAF mutations in melanoma. But that discovery drove drug development programs focused on creating one of the first drugs that was targeted to a mutation, but only in a certain population of patients. So before that, the example of targeted therapy we had was targeting Gleevec and CML, but the Philadelphia chromosome is present in about 95% of CML patients. So that drug was broadly deployed for all of a certain cancer type, not just patients with the mutation, because most of the patients had the mutation. This was a bit of a different paradigm because this drug was actually developed for BRAF mutant melanoma patients. So this is an example of a drug and a diagnostic being developed and brought to market at the same time. So a companion diagnostic for that drug. And again, it's only, so the melanoma patients would be tested for the BRAF mutation if present, then treated with the drug, not tested in the broader melanoma population. Now, why I harp on this so much is because it's really important from the perspective of the broader perspective, not just of the science and the clinical side, but also the market size and the possibility for a drug to be successful for a pharma company to invest in it as they think about, do we want to put our resources into building this thing that may sell more or less of the drug? So heading into this melanoma landscape, we had seen the great success that came with the high efficacy of Gleevec in Philadelphia chromosome mutated CML. And pharma was willing to invest in those drug development programs because even though we were subsetting melanoma patients into half the population, which the business development people are saying that's not great, but we knew that the opportunity to achieve much greater efficacy sort of warranted that investment. So this is the first of its kind where a targeted therapy was deployed in the setting of a sort of basketed population of patients. Similar things were happening with protein biomarkers and other biomarkers, but this biomarker-driven drug development program is really the paradigm is really at the heart of where we can achieve efficacy in cancer. And that has certainly taken root and blossomed in human oncology research. And so we see here the survival curves, just like we looked at earlier, the 3-year survival for melanoma with that inflection point was around this drug development. There's some powerful pictures here. You see a patient that was with metastatic disease that was dosed with this targeted inhibitor and it melted away. We also see an example of before the advent of targeted therapy in melanoma, a cytotoxic chemotherapeutic dacarbazine was used in most patients. You can see the difference in survival curves for dacarbazine patients versus vemurafenib, which is the targeted inhibitor of BRAF here. Okay. So from the mid to late 90s into the mid to 2010s or so, this whole concept of targeted therapy based on discovery of mutations that are used diagnostically, but we can also develop drugs for them. This was taking root in human oncology. But at the same time, the next generation sequencing revolution was boiling to a head, and large-scale sequencing was increasingly more available, and cancer genome landscaping projects were sort of ramping up around the world in individual labs and consortia and beyond. And there was some sense of hope, I think, in the early 2000s that this targeted paradigm, this genetic understanding of cancer would actually lead us to a place where we could find magic bullets for cancer, where we do genetic characterization, we'd find a few key mutations, develop drugs for those mutations. This could help unlock control of disease. But what we found instead was that cancer was much more wildly heterogeneous than I think anybody expected it to be. And that every tumor and every patient, even if it looks the same under the microscope, has a different complement of mutations that are driving the disease and that are potentially sitting there even at subclonal level and potentially able to drive resistance to these therapies as well. That CT of the metastatic melanoma patient, you saw a remarkable response. That patient also ended up developing resistance pretty quickly to that targeted therapy. So here's an example of some of the output of the large-scale genomic analysis of cancers. So if we start looking at thousands of patients, entire genomes, just the breadth of genetic variability across different tumor types and within individual patients with the same type of tumor. This is from a study of several thousand whole genomes in which different tumor types are shown in a column here. in a row, and the rows are different genes, like we've got P53 in this row. And then they're sort of color-coded by, there's a heat map here by what proportion of patients bear a driving mutation in this gene. So if you look across from breast adenocarcinoma to bladder cancer over here on the right, there's lots of different tumor types. But you can just see, just looking at the colors, you can see the great variability in cancers that are driven by P53. It's the most commonly mutated gene in cancer, but there are cancer types where you find no P53 mutations, like pilocytic astrocytoma. There are cancer types where it's almost universal, like osteosarcoma. Over here, there's a great amount of variability in P53's role across different types of cancer. But there's also great variability within tumor types in the proportion of patients with any of these mutations and with a different spectrum of mutations. So we look down the column for breast cancer, and we see lots of P53, we see a little bit of CDKN2A, we see no KRAS, et cetera, et cetera. So again, what this means, and if we were to plot out this sort of heat map as a sort of 3D landscape plot, it's just a total mess. So there's a wild amount of variability within individual patients with the same tumor type, between patients, and even within individual tumors, there's a lot of variability as well. So cancer's complexity arises from evolutionary processes, is part of the challenge we face at the same time. So cancer arises not just in that linear sort of pictogram that we showed at the beginning. In fact, it arises through a branched evolutionary model where over time, these cells are accumulating mutations that end up in different lineages. And then there are occasional selective sweeps. And then sometimes you get these particular clones that will grow out. These can be influenced by all sorts of different factors. So there's a branching evolution that happens in cancer that means that there's a lot of subclonal variability that's sort of brewing in the background that can potentially drive resistance to therapy and creates a lot of different paths to escape treatment, especially chemotherapy. An example of that in a model I'm showing here on the right, based on sort of tracking tumor size over time. Martin Nowak has built this, this was probably 10 years ago, built this model that helped us think about the probability of tumor eradication using targeted therapy, depending on the size of the tumor that we're targeting, the number of cells that are in that tumor, particular mutation rates, and whether there are any pre-existing clones anywhere in that tumor cell population that might drive resistance to disease. So our best case scenario, over here on the right, this red curve, and what we're showing is at the top, this is a diameter of a lesion. So detectable lesions are between about 5 and 10 millimeters by palpation or imaging. That's usually where we'd actually be able to find a tumor. And you can see the number that's mapped to the number of cells in that tumor population here at the bottom. So our best case scenario is this red line, where we have N1 and N2 are referring, we're talking about combination therapy here actually too. So we're already trying to leap ahead to what happens if we can add two drugs together. N1 refers to the number of essentially potentially resistant mutations to drug 1, N2 resistant mutations to drug 2. So if there are 50 potentially resistant mutations to these two drugs in a combination therapy, and there's no mutation that could confer resistance to both of those drugs, our probability of tumor eradication is very high when the tumor is, in this range in here, which is a pretty reasonable space. We've already been able to detect that disease. We've probably found it before we got here. If you find it when it's much bigger, that probability goes down the larger the tumor cell population is. Our worst case scenario on the left is actually monotherapy, so a single targeted agent where there are 50 potential resistance mutations present subclonally when we start. And then we've got varying degrees of probability here as well. This blue line indicates if there's a single mutation that could drive resistance to both drugs, that really impacts our ability to achieve a good outcome. So hopefully this helps show through modeling of mutation rates and resistant clones and single agent therapy versus combination therapy, how the perspective changes on our ability to potentially control disease, and why it is very important to think about how we bring multiple drugs to the table that hit multiple pathways, and what is the background genomic setting in which we're treating these patients. Okay, so this paradigm of our genomic understanding of cancer is an evolutionary process. Again, this has driven a lot of the innovation that has happened in human oncology. Today in human oncology, we have well over 200 oncology drug products. This is a review that spanned 2000 to 2022, and that looked at the sort of FDA approvals for new oncology drugs over that span of time. And since then, the past 4 years, there have been quite a few more. So there are 206 oncology drug products in 573 indications that were approved from 2000 to 2022. This may be hard to read, but this shows you some of the different classes of those 206 products. Targeted drugs are this big slice of the pie here. So these are small molecules that selectively inhibit a mutant protein. There are actually a number of cytotoxics that were still being developed, and even today there are some cytotoxics still being developed. And then targeted biologics, so these are like antibodies that also target mutant proteins by and large. So you see a lot of that is targeted small molecules, some targeted biologics, most of it is targeted therapy. Interestingly though, how many of these were developed in that span of time in a biomarker setting, like vemurafenib for BRAF mutant melanoma? It's actually only 39%, whereas 61% were developed in a biomarker unselected setting. Again, there's a scientific lens on this, and there's a clinical lens on this, but there's also a business lens on this. And part of the reason there's still so much being done in this space is because it's a larger market size. If you start to develop a drug only in a specific biomarker setting, then you're developing your drug for fewer patients, which all sounds horrible that anyone is even thinking about this, but that's actually the way that the resources flow in big pharma. And this will come back to veterinary and why I'm even talking about this in a little bit. Now, are these drugs less effective? I mean, not necessarily, no. It would be interesting to have that analysis overlaid here. I don't know if anybody's done that, but if someone wants to look, I'd love to see what that looked like. Okay, so we have a large and growing human oncology therapeutic portfolio. We also have a large and growing genomic diagnostics portfolio. In fact, over the past 15, 20 years, an entirely new discipline has evolved called molecular pathology at the intersection of genomic diagnostics and traditional pathology. There's a lot happening in this space, is the takeaway from this slide. A lot of mutations are used as biomarkers in standard clinical care for human cancer patients. There are dozens and dozens of companies that are developing genomic diagnostic products, and there are many assays and trained molecular pathologists working in this field. All right, so that's the human picture. Again, there's a lot of need that remains in human cancer management. I think there are some exciting new developments. It does feel a little bit like we may have hit a bit of a plateau in innovation, but from the perspective of veterinary medicine, it's just like a utopia because there's so little that we can do in veterinary oncology. That is starting to change. So we are starting to, in the past 5 years, we have started to get a much better handle on the genomic underpinnings of dog cancers. So I'm showing here some of there are probably about 30 or so studies that have been published in dog cancer using some sort of genomic profiling technique in different tumor types. I'm showing you a few examples here from the most common tumor type in dogs, which is lymphoma. So unlike humans, in the US at least, dogs are not getting breast and colorectal cancer and prostate cancer. A lot of that is due to dogs being spayed and neutered. On the colorectal cancer side of life, that's an interesting question that I don't think anybody has addressed particularly well, but I'm sure is related to predisposition, diet, and a number of other factors as well. But lymphoma is actually the most common cancer type in dogs. So these are 3 different examples of lymphoma genomic studies that have emerged over the past 3 or 4 years, where some type of sequencing approach, exome, whole genome, has been used on tumor normal pairs from the same dog, and we've looked at what mutations occur that are driving the disease. And we have these oncoprints where, kind of like that last one we looked at, different genes are in a row, different patients are in a column, and you can get this snapshot of what genes are involved, what might be co-occurring or mutually exclusive, and what's driving the disease. You'll see across these 3 different studies, there are some genes, if you looked closely or could read it, there are some genes that are shared across the different studies. TRAF3 is commonly mutated in canine lymphoma, for example, which was an interesting finding that's a bit different than the human lymphoma landscape. And then there are a number of others that are for the most part, shared across these different studies, as well as some unique features of disease that have been characterized as well. So part of the reason I show you this is not to get you super oriented to the genomic landscape of canine lymphoma, but to show you that this data is starting to emerge, and we're starting to get a handle on these things. Now, in the human genomic space, it's really evolved into multi-omic understanding of cancer and reclassification hand in hand with pathology. So we are like 10, 15 years ahead, and a lot more resources than we are on the dog side. The dog side, we're still trying to get the basic genomic map. Studies that integrate transcriptomics, epigenomics, proteomics, and pathology are rare to non-existent. So there's still quite a lot of need here, but we're starting to get a better handle on things to at least get ourselves oriented. This is an example of a pan-cancer genomic landscape, different pan-cancer genomic landscape studies where we can compare different tumor types. Again, I'm not going to dive deep here. I don't think we have enough time, but we start to see patterns of mutation. What genes are most commonly mutated across all dog cancers? Well, p53 actually is also the most frequently mutated gene in dog cancer, just as it is in human. Notably, there are some unique features of dog cancers that aren't present in human cancer as well. And there's a lot of rich opportunity there to dig deeper to understand the natural history and development of cancer in both species on where the differences are as much or even more so than some of the similarities. Some examples are canine osteosarcoma, bone cancer, which is often studied as a model for human osteosarcoma, which is a rare disease that is under-resourced in the human space, but it's much more common in dogs. So it's an area where we can learn quite a lot about studying the dog cancer. Human osteosarcoma is largely driven by p53 as well as other mutations. That is true of dog osteosarcoma. But there are novel mutations in dog osteosarcoma that we don't see as much in human, like in the epigenetic gene SETD2. And in fact, in the DMD, the very large DMD gene involved in muscular dystrophy as well, that's mutated in dog osteo. So some interesting stuff to dig into there. And actually, I think I've got a slide to talk about that in a few minutes. Here are some resources on canine cancer genomic data. This is if any of you work in the cancer space, you're familiar with cBioportal. This is the dog version that a group at University of Turin has built. There's also an NIH effort called the Integrated Canine Data Commons that's trying to bring data together. So these are portals like the ICGC and the TCGA. Those various portals are being built for dog cancers as well. Okay, I think I'm running out of time, so I'm going to try to speed up a little bit. I have to mention cats. Sorry, cats always get like the last mention, and that's why Grumpy Cat is very grumpy. We don't have nearly as much information on cat cancers and cats. Mostly targeted studies are what have been done so far, but a little bit of work is starting to ramp up in this space, but there's certainly a big need there as well. Okay, we looked at the human oncology drug portfolio. I don't even have a pie chart for you because we can make a quick little easy table. There are not that many drugs that have been approved for dog cancers. We had an opportunity for our Gleevec moment around 2010 with a drug called Palladia. So much like Philadelphia chromosome being discovered in CML in the 80s and 90s, a mutation was being discovered in a kind of tumor that's common in dogs and not as much in human called mast cell tumor. Though in those tumors in the 80s and 90s, we discovered that many of them were driven by mutations in the receptor tyrosine kinase KIT. That drove a drug development program for selective KIT inhibitors that yielded this drug called Palladia. It's actually a very dirty KIT inhibitor. It hits lots of other receptor tyrosine kinases as well. It wasn't actually developed in the setting of KIT mutations, and the clinical trials did not extensively accommodate that perspective either. And in fact, it ended up being a bit of a dud commercially. Zoetis, an animal health drug company that developed this drug, did not see this as a successful effort, which meant that they didn't put more resources into more oncology drug development until recently. That started to change a bit. But it also isn't really, it is sometimes used in the setting of mast cell tumor. It's rarely used in the setting of the mutation that it was targeting. It is sometimes used off-label as well because it's a targeted small molecule that's very easy for a veterinarian to work with. They don't have to work it up in a fume hood like they would with some of the cytotoxic chemotherapies that they would have to deal with. So it has not been a wild success. So this is pointing to the fact that we don't have a lot of drugs, and we haven't had our tailwind Gleevec moment in veterinary oncology yet. So there's been a lot of ebb and flow. There will be hype around a new therapeutic paradigm. Right now in veterinary oncology, it's checkpoint inhibition, so immunotherapy. That has been very exciting in human oncology, although in fairly narrow responding populations of human cancer patients. That's the current hype train that's happening in veterinary oncology. But there's a lot of concern that dog cancers are not the same as the human cancers that respond to immunotherapy. So we can talk more about that at a future time, but that we've sort of go through the cycle where there's hype and then disappointment, hype and then disappointment. So these are some of the big challenges we face in veterinary oncology. There is honestly a lot more happening in diagnostics in veterinary oncology than there is in therapeutics to some degree. This here, I'm mapping different startups that have developed over the past 5 years that work across dog cancer screening, diagnosis, or disease monitoring. One of these, as Anne mentioned, is a company that I built inside of TGen called Vidium that was doing tumor profiling for dogs to help guide treatment, but there aren't that many treatments. So here you get a sense of what's starting to happen in the genomic diagnostic space. There are a fair number of startups working in therapeutics as well. And of course, there are big animal health pharma companies that have small oncology portfolios. But as an example, oncology for a big pharma company like Merck Animal Health, Zoetis, Boehringer Ingelheim has an animal health component. There are really only 4 or 5 of them. They see cancer as being a multi-million-dollar market at best, whereas parasiticides are billion-dollar markets. So when they're doing their budgets and deciding which teams they're going to fund, who do you think they're giving the money to? It's not usually oncology. It's usually parasiticides or derm or some other area that has a much larger market size. That's part of the challenge that I'm interested in trying to address. So, okay. The point of this slide is to show that there are there is good reason to think that we can look over to what we've done in human oncology based on our genomic understanding of disease and actually lift a lot of that over to dog. A lot of the targets, a lot of the cancer genes that are involved in human cancer are also involved in dog cancer, and they have very high identity. Those proteins have high identity with their human counterparts. They're very similar. They accumulate mutations in similar positions. That's what I'm showing you with these lollipops down here. The BRAF example is a good one. Dog BRAF is actually not often mutated in melanoma because dog melanoma isn't cutaneous typically. It occurs in the mouth because dog's hair protects them from UV. So dog melanoma doesn't often have BRAF, but actually dog bladder cancer often has BRAF, strangely enough. The mutations that occur in BRAF in dog are at an equivalent site to the mutations that occur in human BRAF, but usually in melanoma. So lots of interesting stuff to unpack there. But reason to think that we can look across the species divide and lift learnings back and forth. This is an example of a shared mutated pathways between human and canine osteosarcoma. We talked a little bit about this already, so I'm going to jump ahead. So what are we doing in precision medicine these days in veterinary oncology? In some ways, we've jumped the gun and tried to bring genomic diagnostics to veterinary oncologists before all the drugs are available. Partly how we've tried to bridge the gap of not having control over the drug development pipelines is by virtue of the fact that veterinarians can actually access human therapies through compounding pharmacies and use them off-label. So there are about 10 targeted therapies that are available to veterinary oncologists through a compounding pharmacy. They range from lapatinib, which targets HER2, to trametinib, which targets MEK, to the BRAF inhibitor vemurafenib, but there are issues with that one. So one of the things that we tried to do at Vidium is we tried to leap over that drug development problem, and based on the fact that veterinarians could access these drugs, provide them with information on the genomic underpinnings of the dog cancer, the patient in front of them, and then using pretty strict biomarker rules and the fact that there's high conservation between species, say, if mutation X is present in this tumor in front of you, you could consider, based on this evidence, using this targeted therapeutic, which does this in the human space, even though it hasn't yet been studied in dog extensively. Through that work that we did there, we actually found that here's a survival curve. In blue, we have dogs that received targeted treatment informed by genomic analysis and then were followed over the course of a few years. In red were dogs that received treatment of any kind, but it wasn't informed by genomic analysis. And we found a significant separation between these two curves, but almost more remarkable than that separation is the length of that separation. Out to more than a year. So this is proof of concept that's this idea that we can use genomic information to guide treatment of dogs with cancer actually does have real value and can make a difference for patients. The problem is it's still a very challenging road to walk because it's expensive to do genomic testing. The access to these drugs is difficult. We don't have extensive PK/PD of these drugs in dogs. The oncologists are sometimes skeptical of this approach for good reason. So it is a very challenging road to walk until we have a larger arsenal of therapeutics available for dogs. And then this will be a more viable model. Oh, yeah, sorry. So drug pharmacokinetic and pharmacodynamic activity. So what the body does to the drug, how fast the body clears the drug, and does the drug actually hit the target? Thanks, Carla. So it's all well and good that these drugs are available, but if we're not actually sure if they are hitting their target in the dog, or the dog may be clearing the drug before it has a chance to be very active, then it's harder to know if we're even operating from the right baseline. So I think I'm getting close to the end here. There are we're starting to make some progress in understanding the genomics of dog cancers. There's been a lot of effort on the diagnostic side that's almost leapfrogging drugs. There is activity happening in the drug development space. As I mentioned, a lot of that is these immunotherapies that sort of uncloak cancers called checkpoint inhibitors and then activate the dog's own immune system to attack them. But we still have a lot to do. And there are also a lot of fundamental, fascinating questions that are arising from our understanding of dog cancer genomics. So I'm just pointing to some of these here. I want to point to one example. So bone cancer, osteosarcoma that develops in dogs, it develops in older dogs. So dogs, of course, live much shorter lives than we do, unfortunately. And bone cancer in dogs tends to develop in older dogs. Bone cancer in humans tends to develop in much younger humans. And yet, signatures, like if we were to look at the context in which a particular mutation would occur, like a C to T transition, what are the nucleotides around that mutation that occur in a cancer genome? That gives us a view onto different processes that can be driving cancers forward. So the signature of C to T mutations that occur in a specific nucleotide context is a signature of age, essentially. As cells divide, they accumulate these mutations over time. We can look at those signatures using whole genome sequencing. We usually see those signatures in older, in tumors of adults in humans. They don't show up very often in pediatric osteosarcoma. But in dog cancer, they're enriched in dog osteosarcoma, which it's older dogs, so maybe that makes sense. But it is interesting to think about the difference between, sort of accelerated aging, relative aging, and absolute time and number of cell divisions and mutation rates, and the natural history of cancer in the two different species. I think there are a lot of interesting questions to dig into on this front. There's, as we already talked about, the different mutations that can drive cancer. That's already a fascinating area. Natural history of dog cancers, somatic mutation rates in dogs. And there are implications here for how we manage cancer, not just in dogs, but can we use dogs as a model to ask some really fascinating questions that feed back to human? So one thought in particular is that which I don't think anybody knows that I've seen, is the clonal complexity of a tumor in an adult dog comparable, lower, or higher than a similar tumor in a human? My hypothesis would be that it may well be significantly lower. And what would that mean for if we have a much less complex tumor with fewer potential mutations that could drive resistance? We might have a better opportunity to cure cancer in dogs due to less genetic complexity. Being able to test that alone, if we were able to do a really definitive experiment, that would be a big deal for helping confirm experimentally some of our hypotheses about how these forces impact human cancer treatment. But there are a lot of interesting things we could test on that axis too. So lots of cool questions remaining to dig into on this front. All right, so the last thing I'm going to leave you with is the next thing that I am thinking about doing today is really at the intersection of how do we address the drug development challenges in dogs. We're not doing biomarker-driven drug development in dogs like we are in human. And I've had these conversations with animal health pharma over and over again, trying to convince them with data, look, if you develop a drug in the setting of this particular biomarker, you're going to see much greater efficacy. That efficacy is going to mean that owners will be willing to pay for that treatment, which means that your overall market size is going to be bigger. This is worth your investment. But we don't have an example of where that is true. So it is usually a non-starter conversation with pharma. So can we instead bring to them some novel solutions, some novel drugs and diagnostic tools that can help them with the drugs they already have in their pipeline, but actually expanding the impact of those drugs? And through a circuitous path, a lot of this gets back to sort of evolutionary concepts in drug development that are emerging in the human space, which I've sort of tabulated here, that I think often are involving combination therapies that can help us direct the evolution of a tumor in order to sensitize a tumor to a different drug. If we can bring those therapies over from the human space to the current pipeline in the dog space, then we can be presenting pharma with a solution to their biomarker and efficacy and market size problem, rather than saying you're doing it the wrong way without having the data to convince them. So I think there's a lot of interesting opportunity here to build that sort of unique model at the intersection of really cutting edge science, as well as frankly, a business proposition for pharma, and try to capture some of the resources in the pharma space. Another point to make here is a lot of these approaches that are using novel ways to sort of manage cancer as an evolutionary force face the challenge of you may not actually be nuking the tumor. A lot of how we treat cancer is we just blow it up as fast as we can. But if we really want to manage it effectively, we need to actually be directing evolution into dead ends, or we need to be controlling disease. And that paradigm is still early in the days of development, and it's hard to test. So let's say you have a drug that works with an approved drug by sensitizing a tumor, but it doesn't actually have a therapeutic effect on its own. That's hard to test clinically, because usually the drugs that we're testing clinically are in advanced cancer patients who don't have a lot of time. So you don't have time to sensitize a tumor that's already probably very genomically complex because it's been hit with 10 other therapies in a patient who has a month to live. That's very hard to test. It's not hard to test in dogs with cancer. So I think there's unique opportunity here to build a company that can work on bringing these approaches from the human space and being a bridge to veterinary pharma, but also helping show proof of concept for the human drug development side. So I think I'm over time, unfortunately. So I would love to talk more about that or anything else with everybody. Thanks, guys, for listening.
given space. All right. That was really cool. So on the last slide there, I saw this mentioned, but a lot of this is all about sampling the tumors themselves and identifying mutations in tumors.
Yeah.
What about identifying them before they're big enough to be found? So like cell-free DNA-based assays and things like that.
Yeah.
What's the space there, right? How useful would that be?
Yeah, absolutely. Very
and
useful.
how far developed is it for let's say veterinary oncology?
Actually, it's fair. There's quite a bit of work in that space as well. Yeah. So I totally agree. That's an important part of the discovery work we do, and even the diagnostics that we can build. There are a number of research teams and even startups who are working with cfDNA. And we see a lot of similar patterns to human cfDNA patterns in terms of correlation with tumor size, with various things. Different tumor types shed more or less. We're able to see the mutations that occur in the tissue. We also see them in the cfDNA. So there's a lot of similarities in terms of sort of broad patterns of how cfDNA works in dog cancer. There are also some unique features as well, like the fragment sizes and fragment ends are a little bit different. I don't think anybody's quite figured that one out yet, but yeah, it's an important part of the equation for sure.
Thanks, well, it's great to have you here, and
thanks,
glad
Rob.
to see all this work. So I've got a lot of little things we can talk about afterwards about
Sure.
possible connections for you.
Yeah.
One of the packages that I think might be useful is Cancer Effect Size R, which is an R package. If you're familiar with that one, it tries to control for the size of the gene, the background, the background mutation rate when you try to determine which are the driving genes. And when you mentioned DMD, is that right? What's the X-linked gene that's really huge?
Yeah. DMD is huge. Yep.
Yeah, that's one where that's really important.
Yep.
Have you connected with the Exotic Species Cancer Research Alliance, ESCRA?
No, I haven't.
So Tara Harrison runs that, and if you think dog cancer is hard, the poor zoo vets, you've got tens of millions of dogs in America, and you've probably got tens of rhinosauruses, right? I mean, it's really hard to figure out how to treat cancer in these exotic species, but they've been putting together a database of veterinarians' experience. Like, hey, I tried this and this kind of cancer in this species, just to try to find patterns in there. And you might be able to mine that to find what's robust across species that might be more likely to work in dogs than something else.
Awesome. Yeah, that's a great idea, Carla. Thanks. DMD is an interesting one because what we actually see is disruptive translocations that are also often homozygous, which still has some caveats to it, but is easier to see perhaps it's pathogenic than point mutations.
Other questions?
Yes, I think I'm curious about how has been for you just creating different startups, because that's, I guess, that must be very difficult.
Yeah, in terms of
like the process, the challenges, how has been for you?
Experience in different startups.
Yes.
With challenges in a startup relative to like academia or.
Yes.
Yeah.
Yes, sort of.
Yeah.
Challenges of doing a startup.
Yes.
Of doing a startup, yeah. So the startup world, and as I'm thinking about this new concept, I've thought fairly hard about, I've mostly moved out of academia. I still have an adjunct appointment at TGen, do a little bit of sort of legacy research with folks over there. If I've thought about, do I want to try to spin up a new academic research program to try to tackle this idea of sort of brokering cutting edge therapeutic and diagnostic ideas to animal health pharma. But the reality is the part of the problem that I see that I want to solve is almost a business problem as well. So that's what's led me back to startup instead of academia. But maybe to answer your question more directly, but just to say I'm always actively thinking about this, like why the startup world? Do I want to keep doing this? It is, it's very fast-paced and exciting. I think a lot of people get addicted to the startup life because you're like scrambling to make everything happen, and it's totally nuts, and it fails most of the time. But I mean, that happens in research too. But in research, you often have I don't want to say a more sure footing because I know it's particularly unsure right now, but in a way you're at least at an institution where you have some footing. In a startup, you're often just way out on your own, and if you don't hit your targets, it's all over. So there's something that's, I think, addicting about that. It's not actually, that's not true for me. The startup world is attractive to me because it feels like that's where we're really, like what I said at the beginning, it's where you really get to take a discovery and put it into the hand. You make a product. Most startups are about building a product. Impact, yeah. It's actually getting the thing into the hands of the clinic and where you want to see the stuff happen. So I love that part of it. The challenges are you're scrambling for money. You're not writing grants necessarily, although grants can still be a part of the equation, but you're having to pitch to the people who have money, to venture capital, to angel investors. You're still trying to tell your story and bring in money. So in some ways, there are parallels between trying to get research funded in academia and the startup world. The nice thing about the startup world is the turnaround time is usually a lot faster. So you're not spending a month writing a grant and then it's a year later before you even know if any of it panned out. It's usually like the next day, but you know that it didn't pan out. So that's a pro and a con. It's usually, I don't know, there are a lot of parallels, I think, in some ways between small labs and a startup environment. But the biggest difference that makes me excited about working in that world is that product side, like the opportunity to actually take research and turn it into something useful, or at least try to do that.
Did you get started in this landscape by developing something and then deciding to go the product route? Or did
Yes.
you think, oh, I really want to make something accessible, so let's dream up a product and then... So
More the former, yeah. So the work we were doing to map dog cancer landscapes at TGen, and when we started to see the parallels to human and think about that, how can we put this information to use today? At some point, it started to feel like this has value, and I want to see veterinary oncologists using it. Now, we could have done that through an academic path as well, but we had an opportunity to secure seed funding to build an incubated startup at TGen that sped it all up. So So I guess discoveries, wanting to take them into the clinic, we decided to build a company around it. And then after I had that experience, made a whole bunch of mistakes, had no idea what I was doing, realized that I actually was really excited about the possibilities in the startup space for doing that again. So.
Have you thought about going into glioblastoma at all? I can give you the pitch here.
Yeah, I'll take the pitch.
All right, so Matthew Breen, did you know him yet?
Yeah, I know Matthew pretty well.
Yeah, so when he's a dog genomics person, dog cancer person, and he argues that dogs get about 11 times more cancer than humans, so they can be a really great model also for studying cancer in general. Yeah. But in particular, the brachiocephalic dogs, the ones with the squashed faces, get a ton of brain cancer, glioblastoma. Yeah. And like there's boxers that some of the families of boxers, 25% of them will get glioblastoma. On average, I think across boxers is about 10%, but that's still orders of magnitude more than humans. So we've never seen an early glioblastoma in a human because you only detect it once it becomes symptomatic that you're having epileptic fits and things like that. But in these boxers, you could actually screen them after year 6 and pick up these early glioblastomas that we've never seen before. Yeah. The other part of the pitch here is that there's been over 360 clinical trials in glioblastoma for human drugs, and none of them have worked. Yeah. And over, I think, 95% of them didn't even test if the drug crossed the blood-brain barrier, and which is where we think a lot of them are failing. So this dog model might be great for a disease that's really hard in humans also, and could have a big market impact both in human and dogs.
Yeah, it's a great point. It's clinically, glioblastoma is tricky because it's not very often diagnosed and rarely treated, even by oncologists. But that doesn't mean we couldn't build a program to find it and then do something about it, right? So I think
Matthew has connected with the American Kennel Club, and they have a catchment area around North Carolina that has, I think, 250,000 boxers in it.
Oh, okay, yeah.
Wow.
Yeah.
So,
I mean, I think it's a powerful space where if you achieve success, it would really talk about a tailwind. Like it could be a great tailwind both for the comparative value for human and establishing that model as a pipeline for innovation for human, but also to show the dog world that this kind of approach is useful too. Yeah.
Very nice. And with that, I think we've reached the end of our hour. Thank you very much.
Thanks, everybody.
And join me in thanking Will.